Learning to do bioengineering day 5 was pretty sweet. I did my first genetic transformation where I put a plasmid into some E. coli. That comes later in the day because it turns out that you have to actually prep the cells and make them competent, which apparently improves the efficiency of the basically the ability for cells to take in the plasmid. What I'm doing now, though, is I'm testing that the plates that I poured actually have antibiotic resistance because I failed in doing this last time. So I'm streaking two different strains, one with antibiotic resistance and one without, onto my new plates. And when I go in today, I should expect to see no growth on one and growth on the other. So hopefully that we will see that. Now I'm using this alcohol lamp to do the streaking so that it lowers the risk of contamination just from airborne stuff. I really should probably start wearing a mask. One thing that's interesting is I feel like I'm learning to just be a better, more mindful of sterile technique and what could be contaminated and what not. But so once I did these streaks, I then put stuff in the incubator to let them sit overnight. And now what we're doing is something really cool, which is testing for the stage of growth of some bacteria. So to do the bacterial transformation, you want cells that are in a stage of growth called mid-log. And the way that you test for that is by using a spectrophotometer and look at something called the optical density at 600 nanometers, the OD 600. And basically what I'm doing here is I'm trying to use the nanodrop and use a LB broth blank to set my calibration and then use my actual cells that I've been growing. It turns out that the nanodrop didn't work, so here I'm using a cuvette. It ends up using more of your material, but I actually, since the cuvette should be sterile, I decided to just take those cells so that I still had 5 milliliters of culture. Just so that the number was nice, and I think some of my calculations for like how much calcium chloride to use for the competent cell part of this. Anyway, so now what I'm doing is I'm taking my 100 millimolar calcium chloride stock and turning it into 50 millimolar, which I'll use to do 2 resuspensions of my cells after I pellet them down. And this will basically attach calcium ions, I believe, to the DNA. I need to actually look and start learning some stuff because I feel like just doing a lab is not, it helps you connect more with like what's actually going on, but it's very easy to get trapped in just reading some instructions and not actually thinking about the underlying reasons of why stuff works. So this is a case where I don't really know what the purpose of adding this calcium chloride and doing these resuspensions actually has on the cells. But presumably when I go to heat shock or something, the plasmid is taken in more readily. And it turns out that you have to like put this on ice. I don't even know why you have to do that. But so now I'm just balancing my pipettes, or I will be, I think, so that I can centrifuge them and not have to worry about it. I learned how to use this new centrifuge. I really like this centrifuge. It's smaller, and so I don't have to like use that massive one. But it also has the refrigeration, which is really nice because I think in this case they recommend that you centrifuge at 4C. One thing that wasn't shown in this is basically me writing the protocol. It was sort of taking the instructions that ChatGPT generated, which I don't actually think I have too much of an issue with using protocols that are generated by ChatGPT if it's something that is as common as bacterial transformation or specifically with pGlow or making competent cells, because there's so much documentation online that I would assume it's not really out of distribution for the generations. But what I did is I take that and then I rewrite it in my own words so that I have it in a little document. One thing that I'm hoping to do actually is find a good way to, oh, here I am resuspending the cells that I grew overnight in that mid-log phase in the first calcium chloride. Now we're going to incubate it for a little bit, I think, and then we're going to pellet them down. and then we're going to do the whole thing over again. That's pretty much it. It's just 2 resuspensions. But what I was saying about my lab notebooks is I want to find a way that I can publish my lab notebooks. I'm thinking, since it's written in this document typesetting program called Typest, I might just like copy over the source of my lab notebook every night onto some GitHub repo, and then I can host the PDF somewhere or something like that. Here I'm learning to use this other centrifuge, which also has refrigeration properties, but uses like 1.5 mil Eppendorf tubes. So, just learning to use the equipment. One thing that I do is I like to feel the thing and listen until it's up to speed to make sure that it's not making any weird sounds or vibrating, because I feel like I just really don't want to break any equipment. And I feel like the centrifuge is probably the thing that's most likely to get broken by misusing it. And actually, one thing I did that embarrassed the hell out of me is on the centrifuge that's right next to me, I used it. It doesn't have refrigeration, but it's right there and it's nice to use. I didn't put the lid on it, and so it started making this crazy sound. Nothing bad happened, but I was panicking and I stopped it and whatever. So I think here, I forget what I'm doing here. Oh, I'm starting up the water bath. So that for my heat shock, I was planning to use just a heat block, but someone recommended that I just use a water bath. So that's what I did. And I just dunked them in there. There are these nice floaty things that were cool that made it easy to stick the tubes in. All right, so I don't remember exactly what I'm doing here. Let me. Gotta get more ice. So I think I might be doing the transformation right now. No. Oh, here I was checking it. So I had been incubating this tube. It's liquid media with P-Glow, because I wasn't happy with my results for the plasmid extraction. Even though I used that yesterday, I probably, if this bacterial transformation doesn't work, I'm going to use the plasmid that I extracted today. Well, that's so confusing. I'm recording this the day after this footage was taken. But yesterday, I redid, and you'll see this at the end of the video, I redid the plasmid extraction lab using these new cells, which were, when I pelleted the cells, even in just the normal fluorescent lights of the lab without the UV, it was bright green. It was amazing. So, and one thing that's really cool is that the spectrophotometry data was looking a lot better. So, I am really happy that I did some stuff that was like noticeably made it better. Like I heated the elution buffer up to the proper temperature. I did the centrifuging at the, with the refrigeration. And then I made sure to use this one buffer that had degraded. I found some other ones as good. Here I'm using the flow hood for like the first time, which is pretty cool. I don't even remember. I think what that plate was the transformed cells. So that should glow. And if it glows, then what we know is that we successfully took any B stable cells that don't have the glowing plasmid, and we actually introduced the glowing plasmid, the P-glow plasmid, and that it's producing the protein. So that's, when I go into the lab today, that's what I'm going to be checking for. Okay, so now here I'm starting to redo the plasmid extraction lab, and I'm noticing that I am getting a lot faster at doing these protocols. One, I've done it before, and I'm just more comfortable around the lab, but it really is nice. And I feel like doing these mini preps a second time actually is good, because I can see myself having to do this a lot. One thing that I really want to do is, one of my projects for this summer is to build a bioreactor, and the point of that will be to make it so that I can produce a protein, extract it, and sort of have a little protein factory. Now, we actually have some plasticware or glassware for this purpose, so my plan is to start doing some research on figuring out how we can do this. But there are so many goodies and toys to just play around with. And what's super dope about doing biology in a community lab like this and outside of the confines of, I don't know how it is for maybe PhD biologists, but at least if you're like an undergrad working in a lab, you're probably directed and you're told this is what we want you to be doing, and you don't really have the freedom to just be like, I want to do this. So like one of the things that I'm planning to do is build or at least work on using our OpenTrons. I think it's an OT2. It's not a Flex, and I don't think it's an OT1, but using that to like start, automating some, either making like serial dilutions or just using it as a pipetting robot. And you might be able to see it in the background of some of this footage because it's behind my well, I guess it's in front of my work desk, so you'll see it. Okay, what am I doing here? So we're just doing the mini prep, which is just basically put in a bunch of buffers, wash them out, put in a bunch of buffers, wash them out. So here I'm lysing the cells. And one thing that I had to do that was really annoying this time that I didn't have to do last time is I accidentally sucked up some of the insoluble stuff into the pipette, which is bad because you don't really want any of that junk going into the column. So I had to spin it down again. So here I'm waiting for it to go to 4C. This took forever because I had to look up how to use this thing because no one was around. But it wasn't a big deal. It took a while for it to get to temperature. One thing that it seemed to help is that if you keep the RPM higher, then it seems to drop in temperature faster. I don't know why that's the case. Here I'm using the heat block for the first time so that I can heat up my elution buffer. Heat block's pretty sick, actually. I like the heat blocks. They're fun. I actually really wanted to machine my own heat block. I even did the CAD. I found a drawing for a 1.5 mil Eppendorf tube, and I drew it out, and then I made a grid and subtracted it from a block. The problem is I don't really have access to a CNC machine, and I was looking at how much it costs to order it, and it was gonna be like, I think it was gonna be like $50 from JLC or maybe PCBWay, but it was gonna be like 130 from anywhere else. And the thing is, I don't even want to order that because it's such a simple part that the only reason why I would want to make it is to write the G code myself, or at least write the CAM myself. I'm super pissed that Onshape still doesn't have the. I know it's gonna come soon, but it's like I don't want to use Fusion. I'm done with Fusion. I want Onshape to be my CAD of choice. So here I'm spinning it down again, trying to get all of the plasmid, which is the lysate. So this is another one of these words that you just got to learn the vocabulary. When people say supernatant, lysate, I actually don't know what they mean. I know the supernatant is what's on top, but lysate, I think, is... Lysate contains the plasmid. I guess that's all I know. All right, so a lot of my time is just spent balancing for centrifuging. I'm sure that people don't get as obsessive about this, but the last thing I want is to break one of these centrifuges that I basically get it within 100 milligrams. I think that should be fine. I wonder if any really fancy centrifuges actually have a way to like measure the balance or auto balance, because that would be really interesting. I wonder how you would do that. A lot of this footage is looking at legs. All right, so I think that this is it. I can't even remember what happened after this. Oh, I think the last thing I do is some spectrophotometry to finally get that the data is looking good. So here I'm just labeling, putting everything away. It's pretty slow going. One of the things that I've definitely noticed about lab work is that it would be just great if I could write programs and do like really interesting experiments that wouldn't take, because if I had a robot to say, I want to put my I want to heat shock my cells for 40 seconds, 50 seconds, and 60 seconds, it would be really annoying to do that by hand, but it'd be really easy for a robot to do that. So I'm definitely getting more and more interested in starting to figure out how I can speed up my my work and just make it a lot easier because, I mean, doing this by hand is psycho. Yeah, so here I'm just testing for the purity, the concentration, and of my plasmid extract. The first time I did it, what I didn't realize is that you're actually supposed to wash it with water in between the blank and the. I don't know if it was a in the actual measurement. I don't know if it was a bubble or what actually caused it, but I did this reading twice because the first time I read it, it was like really bad results, but the second time seemed pretty good. I was getting a that whatever absorbance at 260 over I think it's 230. I got 1.9 and they said the ideal is 1.8. So at least we should have some indication and the spectrophotometry peak really did look like good.